be talking about my project using preclinical MR radiomics and machine learning to predict survival for glioblastoma immunotherapy treatment. So just a little bit about glioblastoma and this treatment. Kim went over this a little bit earlier. It's a highly aggressive brain tumor with a very low 2-year survival rate. Current treatment currently involves surgery followed by radiotherapy and/or chemotherapy. What we're looking at is the use of immunotherapies in combination with the chemotherapy drug temozolomide or TMZ, which has shown a lot of promise preclinically and clinically. And the type of immunotherapy that we're looking at is the anti-PD-1 checkpoint inhibitor. So on the right here is an example of brain tumor in a mouse model. The tumor itself is outlined in red, but as Kim said earlier, it can really extend beyond that microscopically. So MRI allows for treatment monitoring via longitudinal characterization of the tumor microenvironment. We can track macrophages after they take up iron oxide nanoparticles, and we can assess the blood-brain barrier using gadolinium. As Anna talked about earlier, magnetic resonance fingerprinting is capable of simultaneous T1, T2, and T2 star measurements, and also creating concentration maps of the iron and gadolinium. So below, we have some examples of iron and gadolinium maps overlaid on a T2 FSE anatomical scan, where we can measure the number of cells and the amount of gadolinium. And then the tumors are roughly outlined in green. So we have a lot of these images. We don't really know what to do with the sheer volume of them. So what we do know is that what Kim talked about earlier is that these images contain a lot of information that reflects the underlying pathophysiology of the tumor. So we could take these medical images and convert them into mineable high-dimensional data and extracting features. And these features can offer information on tumor phenotype and microenvironment, and we can correlate them with treatment outcomes to increase precision in diagnosis, assessment of prognosis, and prediction of therapy. So the first types of features that we're looking at are shape features, which are based on your chosen region of interest, so things like voxel volume, sphericity, surface area. Next, we have first order features, which are based on the image intensity histograms, such as maximum, min, mean, median. And then we move into texture features, which are a little bit complicated and have to do with the spatial arrangement of pixels of different intensities. So as Kim said earlier, tumors are very heterogeneous, and we can extract those features and see if they have any correlation with treatment outcome. So the objective of my project is to develop or utilize a machine learning model that can predict immunotherapy treatment success in the mice based on radiomics features extracted from the images. So the study timeline we're looking at, Kim went over this a little bit, is we looked at 15 female mice injected with the GL261 cells on day 0, and we studied 3 treatment groups with 5 mice each. So the first group was untreated, the second was treated with the immunotherapy anti-PD-1, and the third group was a combination treatment of anti-PD-1 and TMZ. So as you see for the bottom, groups 2 and 3 were given their treatments on a set schedule, and mice were imaged weekly before and after contrast injections. We looked at so imaging consisted of an T2 FSE anatomical scan, and then the MRF each imaging day. So my radiomics and machine learning pipelines, basically what we do is we acquire the maps. So we have the T1, T2, T2 star, and also the iron and gadolinium maps from the MRF. We segment the maps. Our current regions of interest that we are looking at is the brain plus the tumor, so what we identify as the whole brain, and then just looking at the tumor itself. We then input these regions of interest into a program that extracts 107 features in total for each region of interest. This is done using a radiomics package. So these features, 107 features, are extracted for each region of interest for each imaging day. There is a maximum of 6 weeks of imaging, so total of 6 imaging days per mice, depending on their survival. We then want to use these radiomics features in a classification algorithm. So first we identify a binary classifier that determines treatment success, and then use a classification algorithm with feature selection and cross-validation to identify and evaluate features that can predict therapy success. And predictive performance is evaluated using accuracy and the area under the receiver operating characteristic curve. So with preclinical imaging, it's much different than clinical imaging. We have far fewer subjects. In this study, we're looking at an N of 15. So we have some issues and decisions to make on how to organize the data. So firstly, the binary classifier that I'm looking at is, did the mouse survive to the study endpoint or not? So if they did survive to the study endpoint, that is, I deem that as a success, and that output is 1. And then if they did not survive to the study endpoint, that is not successful, and that is given a binary classifier of 0. So as I was talking about earlier, we have some questions before we can actually input these features into a model. So first off, how do we organize the scan type data? We have 6 different scan types, the T1, T2, T2 star, FSC. We also have the iron maps and the gadolinium maps. Secondly is, how do we deal with the longitudinal data? The longitudinal data can vary between mice and groups. Some mice have up to 3 weeks of data depending on their survival, whereas mice that did survive to the study endpoint have 6 weeks worth of data. Next is to look at which region of interest is best. Do we look at the whole brain or just the tumor itself? And lastly is, which features should be included in the model? We have a total of 107 features for each ROI, for each scan type, for each day. So that adds up to a lot of features. And some of those features may be redundant and can be removed from the model because they're not useful. So as just a sort of first go at this, sort of an exploratory process, I use a logistic regression model with a leave one group out cross-validation. And with this one, I just put in all of the features, all 107 features for each day, and compared between the different scan types and the 2 different ROIs. So on the right, I'm showing the results for the tumor ROI, and we compared accuracy and the area under the ROC curve between scan types. We didn't get very good results, around 0.7 or 70% accuracy, and this is using all of the features. So next we want to look at if some features could be removed because they're adding bias to the model. So this is where feature selection comes in. And this was, did a study with random forest feature importance. So basically this ranks features using an impurity-based random forest model. And as you can see, even between scan types, the feature importance varies between the scan types and also between the 2 different ROIs. So features lighter in color are more important, are less important, and features ranked in darker colors are more important. So there is quite a variability between the scan types and the ROIs themselves. And what we found was a lot of variation between the types of features that were deemed most important for the different scan types and the ROIs. A lot of texture features were labeled quite important. And then some shape features and some of the first order features as well. So with feature selection, this reduces overfitting of the model, improves accuracy, and overall reduces training time. So some methods I'm looking at is we can remove highly correlated features. So on the right is a Pearson's correlation map between features. So features that are highly correlated can be removed because they offer redundant information. There are also filter methods that we can look at which select features based on their relationship with the target variable, so the outcome variable. There are also wrapper methods which search for well-performing subsets of features. So basically assessing different numbers of features and which produce the best performing model. So some of the classification models that I'm looking at are first was a logistic regression with a leave one mouse out cross-validation, and then different feature selection techniques for that and comparing between the different scan types and between the ROI types. With this, we didn't get very good accuracy or area under the ROC curve. So something else that I'm looking at is what's called an XGBoost classifier. So this is a model that has performed very well in Kaggle competitions. So it's an implementation of a gradient boosting decision tree algorithm. And I'm currently working on hyperparameter optimization for this model, and then further including feature selection to improve training time and improve accuracy. So just as a summary, the preclinical data is very multi-parametric, and the longitudinal nature of it poses challenges in organizing the data for model input. We also have feature importance that varies between the scan type, the ROI, and the treatment time point itself. We're looking at feature selection techniques that will be used to remove redundant features from the model to improve accuracy. Also looking to test and compare different classification models. So currently the logistic regression and the XGBoost classification. If there are also some other things we can look at if we want to assess which features might be early treatment or early identifiers of treatment success. We can look at just early treatment time points in the model, although that does pose a challenge as it does decrease the amount of data that we're working with to test and train the model. So with that, I'd just like to thank my supervisor, committee members, lab members, and our sources of funding, and I can take any questions.